All right, hello everybody. One more lecture, Biol3157, Bioinformatics and Functional Genomics. So I talked briefly in class, had a nice discussion with you guys on Friday. This is just a few slides to try to put that down into a video format for you. So we were talking about population genetics, and we were talking about sequencing a set of samples and then getting a matrix, which is the genetic variation, SNPs, by samples. Um, so basically, we want to start investigating how these samples are related to each other. And there is no quantitative number that describes exactly how they're related to each other. You have um, a sample by sample distance matrix, which comes from the sample by variant genetic matrix. And we're going to try to summarize that into a lower and higher level. Usually samples that are nearly identically related to each other. Sometimes you get two samples from the same individual or two plants from the same mother plant. They're quite closely related. We'd like to cluster those, clump them together as biological replicates. But as you go up this sort of relatedness tree, you get to the ancestral population. So that's the assignment today, and you made it partway through that last week. The concept is that you're going to go through this quality control. We're going to go through a filtering process of both excluding bad samples and then excluding bad variants, which usually have failed and you don't have a lot of information from them. And then you go around this iteration filter one more time, and now that you've got good data for most of the samples and most of the sites, the nucleotide sites, you can start to prune it back and get some information about these samples. And often there'll be a few outliers, like different species, or a few samples that are just mistakenly put into the group, and they're not really comparable. And then a whole bunch of variants that describe those. So if you go through one more round of filtering out outlier samples and the rare variants that come with them, you'll get a final genotype by sample matrix that is comparable to a population, often within a biologically reproductive species group. Now, when we're talking about variants, I just wanted to remind you that most variants are rare. We all have private variants that we're born with, and if we pass those on to the next generation, they can become polymorphisms. And over time, if they remain in a population for a long time, large populations there's a better chance of them being reproduced. And small populations, they can stochastically go extinct. So this is a simulation, and you can see what happens to variants, but eventually they can, even when you start at a common allele frequency, they'll go out just by drift. But most variants start rare, and so that's what we'll be dealing with. If they're very rare, as in one individual, they're not going to be informative about relatedness. So we usually filter out those singletons. Now, we're going to be switching back and forth between whether we're interested in learning about the samples, using genomic data to describe the samples, where they come from, what they look like, and that's going to use all the whole genome data together. But then we'll flip around once we have a set of samples that seem to be from a more or less randomly mating population. Then we'd like to look at those variants, go after the genes, and start scanning for functional alleles. And when we turn the matrix to the side and scan down the SNPs, we're assuming that these SNPs are now behaving more or less independently across samples because they've been shuffled through history, and recombination will separate them until you get down to the linkage blocks. So that's the switch between looking at sample relatedness and then looking at how SNPs are related to each other and to the other traits that you might have. So we're in this era of next generation genomics, moving from second gen short reads to third gen long reads. And the old days, we tried to build up a genome by linkage maps. We didn't really have sequences, and genetics was king. And now we do genomics first and think of your specific gene lists second. And it doesn't mean that you come into it completely blind. A lot of published work and decades of research have given us lists of very important genes, and we might want to put those uh, down ahead of time and then go through the genome and say, are these genes that we had a priori information for as being important, are they enriched in any ways? Do they show interesting patterns compared to all the other genes? But nowadays we sequence the whole genomes and we look at all the genes, and you like to compare the outliers to your list of likely candidates and see if that overlaps in any way. If you had a good guess, you would see the genes, your a priori list, standing out in the back of the genome. And if not, maybe there's something wrong with your analysis, or there's something wrong with your gene list in the first place. Okay, so for bioinformatics, this is a rapidly moving field, and I just bring some old slides to try to put it into context. Mostly we're dealing with sequence analysis. There's other types of bioinformatics that deal with image processing, scanning, brain scans, for example, is a huge area, computer vision, but today we're going to be talking about sequence analysis, and that's almost entirely DNA sequence analysis, or RNA sequence analysis would be the expression levels of genes, and then you can also do protein level analysis, often from the translated DNA sequence. So you want to understand that there's a lot of tools out there, and getting the best, most current versions, ones that are stable, actively being developed, can handle large data sets, recommended by others, that you can usually build pipelines of multiple software tools. Use one to process the low-level sequence, even base calling software then gives you sequence. You might do read mapping with that sequence and variant calling with that sequence, and then you get to this SNP by sample matrix, which is the starting point for our lesson. Do you build your own software tools? Rarely. We do scripts usually and wrappers, one set of parameters that works with data that's relevant to you. Experimental design is a really important part of bioinformatics, so we don't spend enough time talking about the layout. And this requires consultation with the biologists, understanding their questions and helping them choose really important sample design, make sure that they're sequenced adequately, and that you have technical and biological replication in there and outliers, like usually it's a plate format, so you want to have a few empty wells and some outliers in there. And so the experimental design will be in full force. The transfer of concepts from other disciplines, and to know that it's not only a technological and mathematical approach. We need a lot of soft biology and subjective inference. And then you want to be able to say, this is what's been done, but where are we going? And have a good idea about long reads coming next, and then be able to do sequencing in the field and distributed kind of workflows. Those are all the future approaches. And so how does the one experiment and pipeline that you're building today, how is that going to change in the future? You want other people to use what you've done. And don't hesitate to get the defaults and look at those defaults and question why are they what they are. A lot of times the defaults are set for somebody else's experiment, and you need to know if they apply. So take your time with that. So for this section, we're trying to understand genetic relatedness at multiple levels, at this kind of species or subspecies, the populations, the ancestral populations that would have been more or less random mating. And then we'd also notice that there's probably some more closely related individuals that might be from families or even clonal lineages, multiple branches on the same tree. And then there's admixture, which is recent introgressions between populations that have been isolated in the past. And you can distinguish recent admixture from ancestral population structure by looking at blocks, by going down the chromosomes, looking at patterns of variance, and in a few generations, those blocks will be still quite large. And those blocks are defined by recombination events. They're known as haplotype blocks. There's multiples depending on the number of founders of populations. And all of this is pretty straightforward. It was theoretical for a long time, but now that we have high density markers, we have more markers than recombination events, and so you can really see these blocks. And it makes it easy to order the markers based on recombination rates. So private variation we talked about briefly already, and also these polyploids. So sometimes, especially in plants, you don't get a mother and a father that then recombine their chromosomes into the offspring. The parents double their chromosomes and fuse, and then all the offspring are identical. So sometimes you get variants between those parental genomes, but they're not segregating, and those are known as homologous variants. We won't deal too much with that today. So let me come back to experimental design for a minute. Sample selection is really important, and we can have many barcodes in a single lane. That allows you to demultiplex after the sequencing and figure out which sample that particular read came from. So there's really no limit now to the number of samples that you can put in, and we're often getting whole genomes on hundreds of individuals in a single reaction for a few $1,000s. So how we step up from there is becoming a computational challenge. We'd like to identify more alleles as well as their frequency and determine the extent of population structure. And when you're sampling now, we're talking about geographic sites. Where do you sample? And if you want to identify population structure across a range, we'd like to usually have quite a bit of geographic breadth. That's not a bad starting place. Of course, the best starting place is just what you have in hand. And so if you can do a search online and email people and have them send in samples, and you can process the DNA, that's a good way to get your sample breadth and geographic breadth up. You want to make sure you have that outlier sample. You also want these maternal line replicates, maybe 2 leaves or 2 branches from the same tree, some in group. And if you have a rapidly cycling plant where you can grow up seeds and get seeds from those plants, you can do family level analysis. Sometimes the maternal lines are all faithfully inbred, and you can get multiple replicates that way. So you basically want to have your balance between geographic breadth, some known genetic outliers, and some known closely related individuals. And then you can see how your genetic matrix compares with your prior. And you'll be able to weight the relative genetic distance to the outgroup and to the in-group, because all of these genetic distance calculations are relative to the samples that you have. So it's really important in the experimental design to put in, you may have other outgroups that you didn't know were outgroups in your sample. You may have other in-groups that you didn't know were in-groups, and then you'll be able to believe those results in comparison to your experimental design. I hope that's clear. That's important. Sequencing depth, well, we have 2 chromosomes usually in heterozygous individuals, unless you're dealing with these inbred weedy selfing species. So you'd like to get probably 2x coverage. Even 2, 3, 4x coverage is good for having confidence in identifying those frequencies. It used to be kind of prohibitively expensive to do that, but now that's known as low coverage. Anything sort of less than 10x is low coverage. And you'll be able to impute a lot of missing data because of the relatedness within families and even within populations, especially due to linkage disequilibrium. So the coverage can be rescued to a certain extent by the relatedness. And the budget is, of course, something that you have to work within, but it's getting easier and easier these days. So that's The way it works is pick your favorite organism, go out in the field. So we used to, this was one of my PhD students from Chicago. We spent a lot of time in the field just developing these tools and working it out. And now the scripts are really, and the molecular approaches are really easy to apply at any level. You'd like to understand something about the environmental heterogeneity as well as the geographic breadth. And so a lot of times there's this habitat level. If you're in the forest or a sand dune, it might affect the genetic diversity, and it certainly affect the species abundance. And if there's a few kind of shade adapted types, they might be filtered into the forested region, and those kind of questions you can start to answer. Now these sand dunes actually evolve on the scale of the plants themselves. They hold the sand, and where there is no sand, the sand grows up to 30 centimeters a year and can bury trees in certain cases. So what we found from this first study was that there's quite a bit of variation even within these little sand dune plots. Some genetic variation that goes back to the last ice age with the square and the circle, or the square and the triangle group. We found two different subspecies, well, two gene pools within each of two subspecies was really kind of surprising to see. We're all caught really in this little refugia between the east and the west, and the north and the south. We've also done some more work in Pelargonia, Australian Pelargonia, and you should look up a paper by Adrian Nicotra and Borbitz is the last author. We published some for the first studies out of the ANU lab recently. All right.